Hello, everyone. Thank you so much for joining us today. My name is Stephanie Cristello. I'm an artistic advisor to Expo Chicago, a critic and a curator who lives and works here. And it seems like it was just yesterday, though it was a while ago when I was artistic director for Expo Chicago, that we had the gleam of an idea for a symposium surrounding the very topics we'll be covering today. And so I just wanted to say how proud I am of the whole team at Expo for really turning this idea into an amazing series of diverse discussions, events, programs, and screenings that are available at program.expochicago.com. So the topic that we're covering today is called On Viruses, Critical Theories of Contagion. And I wanted to give a little bit of a background of how we'll be talking about what is, for everyone, a very difficult topic and something that has permeated our daily lives for longer than we could have imagined at the onset of the COVID-19 pandemic. In terms of viruses, I wanted to give a little bit of a background of sort of how we'll be approaching this as both a metaphor and a reality for our current moment. One of the largest sort of overarching themes that connects the artists and practitioners who I'll be introducing shortly that are joining us has to do with contagions and its constructs. So looking at the biological, social, and also natural relationships that we have to living with not only viruses, but the micro overall. I've been thinking a lot, and we'll sort of get into this during the group discussion about practices of medicine and healing and how that has changed so much over the centuries. And one of the sort of key topics that we'll be covering also is parallels to medieval practices, thinking about the harm as also the cure, the killer and the saint, as it used to be called. And so the purpose of today's conversation is to really think about pandemic as a form or thought process through which we can access each of the artists and practitioners' practices today. So joining me are going to be Sean Raspet, who is an artist that is currently on view in the Sustainable Societies for the Future exhibition that I co-curated with Kirsi Junge-Stevensborg at the Malmo Art Museum, and is a project of the Expo Chicago Curatorial Exchange with Denmark. Hương No, who is an artist who has done multiple works that couldn't be more perfect for this moment. I had the pleasure of being introduced to her work through The Scene, where we published a long-form essay by contributor Noah Hénaff, and whose work is currently on view at the MCA Chicago, which you can all read, I believe, on the program.expochicago.com website, as well as thescenejournal.org. And Elizabeth Hénaff, who is a professor at NYU, Dr. Elizabeth Hénaff, and whose work with viral organisms and microbes have been really influential in terms of the thinking about all of these topics. And so before we get started, I wanted to thank our presenting media partner, Ocula, who will be publishing a full transcript of today's conversation as part of their conversations series. So we're really excited to be essentially writing this live as we try and navigate the thing that has been consuming us all. So without further ado, I wanted to hand it over to Sean Raspet. And then after each of the artists here give a little bit of a presentation, we'll open into a group discussion and have plenty of time for Q&A should any of the audience have questions between now and then. All right, off to you, Sean.
Thanks very much, Stephanie. Let's see, just waiting for my screen here. Okay. Yes, I'm just going to do a little run through of some recent works that might be kind of relevant to the topic. So this is called Screen, and it's a It's a work I did in collaboration with Chiara Eldridge. She's a researcher at Johns Hopkins, or previously was at Johns Hopkins. Now she's in Seattle. Basically what this is, it's as you can see, a kind of pixel grid that's made up of, within each of those little vials is a media that's growing human stem cells, what we call retinal organoids. So what that means is it's basically a kind of retinal stem cell. And it starts as an undifferentiated stem cell, and through different kind of chemical signals, it's made to kind of turn into a retinal stem cell. And so I guess in a nutshell, this is kind of, in a way, kind of a reversal of a typical screen where you're looking at a kind of pixelated grid that projects usually 3 different wavelengths of light, 3 different colors. And this is a kind of pixelated screen of human cells that receives those 3 different colors. Next slide. Okay, and then also related to kind of receiving and receptors, this is a exhibition called Receptor Binding Variations. Each of those kind of little boxes on the wall is a scent diffuser, and there's 8 different scents that I created for the exhibition, and that's the kind of the form of the exhibition. Each of those scents is designed to, as much as possible, target a specific human olfactory receptor. So that's a receptor in the nose that is what allows you to smell. And we have about 400 different types of olfactory receptors. So it's a lot more than in vision, which just has 3 color receptors, plus the kind of rod cells. We have 400 different types, and each type is specific to a range of molecules, a range of kind of molecular features. So it's a way of understanding the shape of a molecule that you smell in the air, or a complex mixture of molecules. Basically the scents in the diffusers are designed based on the molecular shape to target specific olfactory receptors. And I guess as a parallel to today's discussion, that's also, in a lot of cases, what drug manufacturing does. So they design a new molecule that targets a specific receptor in the body and causes some sort of health effects downstream from that. And that's a little bit different than a virus vaccine, but it's a parallel kind of enterprise. Next slide, please. And then so related to new molecules, this is a new scent molecule that I developed, and that was synthesized in collaboration with Shengping Zheng, who's a professor at Hunter College. So basically, I've been looking into the kind of molecular shape and then how that corresponds to scent for quite a while. And based on that, I've kind of designed, I guess you could say, different molecules that I thought would have an interesting scent, and a new kind of scent that hasn't been smelled before. It's a little bit like, in a sense, developing a new color, because almost every molecule that you can smell, each different molecule will have a different smell. And so this is a molecule that hadn't existed previously to us synthesizing it, and so it has a different smell. Next slide, please. Somewhat related to that, and going back to a little bit to pharmaceutical development, this is a project in collaboration with Francis Seng. And basically what it is, it's a website, matter.farm. And what it is it's kind of a ongoing AI generated, or machine learning really actually generated set of pharmaceutical compounds, potential pharmaceutical compounds. So all of these molecules, didn't exist previously, and this kind of AI is just going through and spewing out new ones, basically. And then it matches them to what receptors in the body they may target or inhibit. And so again, that's how a lot of drugs work, is they bind to a receptor. Receptor is kind of like a switch on a cell. You could think of it that way. And they turn it on or off. Next slide, please. Okay, and then this is it's kind of from a ongoing series of works related to mutation and kind of biological variation. These are some morning glories that have been, basically, they've been developed by exposing the seeds to radiation and then which causes kind of randomized mutations and then breeding those through a process. These ones were developed by the Institute for Radiation Breeding in Japan. And there's 2 different varieties that have been put together in proximity to then crossbreed for this particular, I guess, installation. And in addition to this, I'm kind of working on a longer term project where I've been exposing seeds to radiation that are related to a kind of native forest ecosystem. And I'm kind of planting a forest with the resulting seeds here in the US. And that's, a lot of people might have a connotation of mutation as bad or unnatural, but it's a process that occurs, throughout evolution. And it's, in this case, it's a way of speeding up evolution as we have a kind of climate that is very rapidly changing. Things like plants and animals have a hard time adapting in time to these kind of changing conditions. So speeding up the rate of mutation can actually have a beneficial effect on the overall ecosystem. And that's it. Thank you.
Hi, everyone, and thank you so much to Expo for hosting this, and Kate and Stephanie for organizing, and thanks, Stephanie, for that introduction. Thank you, Sean, for your presentation. That was really interesting. My name is Hương, and I'm an artist based in Chicago, and my research-based practice is oriented by a desire to understand how difference and belonging is constructed, performed, and enacted, and how I might find within that discourse sources of resilience and resistance. After the 9/11 attacks, I wanted to understand how a culture of fear was being shaped by our political leaders who were mobilizing and racializing the term terrorist in order to defend aggressive military interventions into the Middle East, to initiate a sweeping surveillance program that drastically infringed upon our privacy and civil liberties, and to legitimize the detention and torture of suspects without habeas corpus, and of course, to further line the pockets of the already inflated military industrial complex. So this was the work that I was making at that time, which is a collection of handmade objects that reflect on the type of consumption that was encouraged as a solution to protect oneself from biological warfare, rather than doing the difficult work of diplomacy for the collective good, hence this title, Escape. And this rhetoric of fear did not remain in that time period, but instead has pervaded US culture and has normalized itself within the Trump administration and popular media. Next slide, please. Escape took visual cues from a government readiness campaign, which you see here, and what was then the beginning of a fringe prepper movement. The work has been described as prescient, but for me, it was simply a mirroring of what was already happening at that moment. From my perspective as an immigrant, as a refugee, an Asian American woman, someone who has been put into that category of the other before. Next slide, please. Now, what is that history of the othering of Asians in American culture? While the category of whiteness was opened up to most Europeans by the end of the 1800s, this was the first time in which immigrants in the United States were treated differently based on ethnicity. Officially beginning in 1882 and continuing until 1943, the Chinese Exclusion Act restricted Chinese immigration based on their profession, familial relationship, and the first time that a photographic ID was required of applicants, which was then a new type of surveillance technology. And working in tandem to shift public opinion was media representation, in which Chinese immigrants were caricatured and presented as irreconcilably foreign, criminal, greedy, and diseased. Chinese immigrants, like the images that you see here, were represented as eating rats, as being associated with diseases like cholera and smallpox. That was a very common belief. But even before the Chinese Exclusion Act, the Page Act was passed, which excluded only Chinese women who were assumed to come to the United States as sex workers carrying and spreading venereal diseases. So this association between Asians and disease has long been a part of defining whiteness or non-whiteness and hence belonging in the United States. Unpacking this history is essential to understanding the racialized economic and political foundations of the United States. As Lisa Lowe has argued, Asians are seen as the perpetual immigrant, as the foreigner within. Next slide, please. The character of the immigrant as easily vilified laborer or model minority is a barometer for how the nation wishes to define itself in that moment, and it serves as a buffer to control more volatile racial unrest. In order to prove their family relationships to citizens, Chinese immigrants on Angel Island in that time period had to undergo grueling interviews, often lasting for hours or continuing over days. US officials, hoping to deport as many immigrants as possible, posed obscure questions about their villages and family histories that interviewees would have trouble answering correctly. Next slide, please. So compelled by what these interviews might tell us about these invisible constructs of national identity, collaborator Hong An Cheong and I created an ongoing performance that integrates questions from the years of Chinese exclusion, from current naturalization tests, interviews for visa and green cards, and more recently, questions posed to airport detainees during Trump's executive order 13769, more commonly known as the Muslim ban, and then also most recently around COVID. Within the span of this performance, we choose a random audience member for fictional citizenship who undergoes the actual process of being interviewed for citizenship. But all people there are witnesses and imagine how they would answer these questions. Within that span of the performance, the project problematizes citizenship as performance and the notion of allegiance and structure of power implicit in the state. Next slide, please. This is the current installation at the MCA, and we had planned to do another iteration of the performance tomorrow over Zoom, but that might change because of the unfortunate recent layoffs at the MCA. Next slide. This painful history of anti-Asian sentiment has been brought to the fore more recently with anti-Asian racism encouraged by former President Trump and his administration. But what is interesting is that journalists like the one here in this article, What's Spreading Faster Than Coronavirus in the US, Racist Assaults and Ignorant Attacks Against Asians. What's interesting about this language is that journalists have been using this metaphor of disease to talk about the spread of racism and misinformation. And while that creates a powerful rhetorical device and a mental model, the language ignores the fact that our culture is built on this racism against Asians and our contingent belonging. In other words, the racism has been there all along. So what I believe is that understanding how these ideologies are constructed and deliberately spread might help us generate strategies to resist and subvert them. Thank you.
Hi, everybody. Thank you, Sean, for your presentation. Thank you, Huang, for your presentation as well. My name is Elizabeth Hénaff, and I'm a professor at the NYU Tandon School of Engineering, where I lead the Laboratory for Living Interfaces. And at my lab, we are interested in studying the continuum, the microbial continuum between organism and environment. So looking at the continuum between the human microbiome, the environmental microbiome, and how those two interact with each other, particularly as a function of our design decisions that we make for the engineered environments that we occupy. I think that when we're thinking about contagion and theories of contagion, there's certain threads that are interesting to pick up with respect to that. One of them being that I see contagion as being a breach of boundaries. You know, so we feel like this external biological agent has breached the boundary of our body. And one of the things that we've been really thinking about is how the boundaries of our bodies are actually porous, and they're there's a constant exchange of biological material between our bodies and the environment that take the shape of these changing microbiomes. Another one is that contagion, when we're thinking of disease, it feels like a breach of identity. Like suddenly, our biological identity has been hijacked by this contagious agent, by a virus that's injecting DNA into our cells or RNA into our cells. And I think that we can also learn from our understanding of the human microbiome that actually, this feeling of the human identity being in isolation or being unique is actually not the case. And so in our identities, especially our genetic identities, are actually a complex makeup of the genes that we encode as humans, but also the genes of all the microorganisms with which we live in symbiosis. And so part of my practice is of scientific research, but what I wanted to talk about today was a few art projects that I've worked on recently. And in particular, in my art practice, I'm interested in thinking about interfaces that we have with the invisible biological component of our environment. Because ultimately, when we're studying something that's invisible, the only way that we can interact with it is through certain objects, instruments, tools, and those objects are what dictate our relationship with it. Next slide, please. So this is a project. This is a series of artist books that I made entitled Gene Zines, and so each one of these in the series is an interface with the microbiome of a particular environment. This particular edition was an interface with the microbiome of the Gowanus Canal, which is a Superfund site here in Brooklyn. So this Superfund site has been polluted with industrial contaminants over the last 150 years. This is, only the latest in the violences that this land has seen, given that it was previously occupied by the Lenape people, and the Dutch settlers both displaced them and also the ecosystems that they relied on. But the way that this interface works is that the paper is embedded with DNA extracted from the microbiome of the Gowanus Canal, which happens to, over the course of the last century, have adapted to this contaminated environment and encode metabolic pathways that allow it to degrade the toxic compounds that it was challenged with. And so this gene zine is a genomic note from the underground serving the subculture of multi-species collaboration, in which you can detach the piece of paper that contains those DNA molecules and embed it in a site in need of remediation, where the local endogenous microbiome can absorb that DNA and use its sequences. Next slide, please. This is a project that was in collaboration with the architecture studio The Living, that is directed by David Benjamin and Kevin Slavin, who was a professor at the MIT Media Lab at the time. And this was a commission by the Storefront for Art and Architecture, a gallery in downtown New York City, in which they asked us to think about what would it look like on an architectural scale if we were to design for microorganisms. And so this is kind of a gymnastics in scale, because microorganisms exist at a microscopic scale, and we exist on the scale of a meter or for our built environment. Next slide, please. And so what we did was redesign the facade of the gallery using bioreceptive materials. So using both materials and design that was aimed to provide amenable microenvironments for microorganisms to be able to inhabit those spaces. And this was paired with also a scientific DNA-based study in which we studied the microbiome of those panels that we had designed, as well as the adjacent concrete surfaces. Next slide, please. So just as I'm interested in bridging bridging across biological scales in space, as with the previous project, I've also been interested in bridging across biological scales in time. And so this project was a series of tide clocks that I made. So this one was installed in the Hudson River here in New York. And so what happens here is that this buoy fills up with water at high tide, and then as the tide recedes, the water is maintained in the buoy structure, and you can visually see the volume of water that was displaced by this kind of more than human clock system. Next slide, please. And this was an iteration that was installed in Okayama for the exhibition If the Snake. And so this particular installation was paired with a metagenomic study of two different bodies of water, one of them being the river that you see here, and the other one being a moat, so a closed body of water that was around a castle. And so that metagenomic study showed that the microorganisms of those two bodies of water had been related a long time ago when the river water was used to fill up the moat, but since then have followed very different evolutionary patterns. Next slide, please. And so ultimately what I would like to kind of put forth for discussion for us together is this idea that we have never not interacted with the invisible biological world, but the tools and the interfaces with which we have to interact with that really dictates our relationship. And so on the left here, you see a clinical swab, which we're all way too familiar with these days after having one, uncomfortably stuck up our noses for COVID testing. So that's a tool that's an interface to interact with the invisible biological environment. And that tool has a particular valence, like it feels like clinical, and it gives us a sense of contagion and of disease because it is, a clinical instrument. And the image on the right is also just as much a collection of a microbiological sample. There's, billions of microorganisms in a handful of soil, but somehow the image on the left has a different kind of feeling to us. It has these connotations of connection and groundedness, earthiness, healthiness. And so I think that in these times where we're all extremely aware of this invisible biological component of our environment, it's interesting to think about how the objects and rituals and other types of interfaces have manifested what is a very new relationship to that and for all of us. And I think that's it. Oh, yes, and all of my projects are highly collaborative, and so I have many people and institutions to thank for all of these.
Thank you all so much. So we're all going to come on camera now for the discussion. I'm really digging into what is a wealth of information here. I'm sure for all of the audience to process, even for us as well. I know that some of you have worked together in various capacities before, but even just seeing the breadth of the projects over the few years I've known some of you has been really inspiring. And so what I appreciate about all your work is sort of this ability to take the structures that surround us that are mundane, banal, or even, as Elizabeth said, these invisible structures, and make them very real and present in new ways. But what I wanted to sort of start with is to take a step back and ask maybe a mundane question, which is how you all relate to time in your work. And what I mean by that is whether or not you see some of these projects as historical reactions, as speculative futures, as even proposals of potential solutions that could be brought into society or science or innovation in certain ways. Where do you feel like your work falls upon on the time spectrum? Like, what time zone are they in? And maybe, Sean, we can start with you.
Yeah, I guess there might be a few different maybe time spectrums, kind of depending on the work and other things. But I guess what comes to mind with for example, the kind of forest project where there's it's all kind of species that would be in an ecosystem together, that are then the seeds are exposed to radiation for mutation, and then it creates a kind of, I guess you could say, more rapidly evolving ecosystem. And to me, yeah, that is something that I think about, is that kind of scale of almost like evolutionary time in a which we are which you can see in observable time over the course of seasons with plants and things. So yeah, but definitely this kind of, and I think along with time there's an idea of change, and an idea that nothing is static, and what we think of as a species is not like, it's not even a real thing necessarily. It's a kind of a bit of a construct, and it's just a collection of organisms, that, and they all kind of change over time. And so yeah, thinking about those kinds of, I think there's a kind of porosity between organism species, certainly in the ecosystem, that I think kind of aligns with what Elizabeth is talking about in terms of the porosity of the body and the kind of microbiome and the surroundings of of everything, really.
Yeah, and I just wanted to make a remark too, because I don't think it's been explicitly said yet, but these boreal species that you are irradiating in order to introduce biodiversity, the goal, from what I understand it, is to basically introduce more varieties of plants in adaptive evolution that would be able to withstand the effects of climate change and boreal forest dieback, right? Which is what many climate scientists call like the tipping point. Once the boreal forests die, the CO2 emissions that would snap out of that, with the inability of these boreal forests to clean the air, would basically take us to a point of no return. So in some ways, it's almost avoiding the time of no time when Armageddon would happen through trees or something like this.
Yeah, it's maybe keeping, I mean, this is, that's obviously like a theoretical and, grandiose goal, or, that would be a great outcome, is to in some way bolster the kind of boreal forest ecosystems. And this project is, yeah, obviously a small scale kind of move towards that. But yeah, it is something that is very much born out of thinking about those kind of these rapidly changing ecosystems. And really there's a point where things are kind of slow, and then there's a sudden collapse. And trying to avoid the collapse is through a kind of acceleration of the change in mutation in the present. I would say that's the basic underlying premise or hope, for what I'm kind of experimenting with there.
Yeah. And Hung, I was, shocked and relieved at the same time to publish the text on your work, Escape, which you covered a bit in your presentation at the beginning of last year. We published it in, I want to say, April, but the text had been in formation for a few months with the writer in advance of that. And it was, in some ways, you told the future in this work. And so I'm wondering if you can talk a little bit about, how your perspective has shifted now, knowing that, obviously you're tapping into these long histories that have a habit of unfortunately reappearing, that there are these cycles and typologies that have stayed past the course of time. But yeah, I'm wondering if you could speak a bit about that.
Yeah, I really appreciate the question about time. Thanks so much. Yeah, I think of my work as being archaeological and futuristic because that's what we do as humans, we are able to look at the past and help us understand the present, hopefully. But we are also able to think about the future. And for me, I'm constantly holding those two in relationship to the present. So I'm always thinking about how they relate to the moment that we're in order to pull out what story needs to be told right now. Because otherwise I would just be overwhelmed with the kind of potential of all of the interesting stories that can be told. But yeah, for escape, like I was saying, I mean, I felt like I was really just reflecting the present, you know. There was, of course, an element of dystopia in it, which is always a kind of a projection into the future. But for me, I felt my job as an artist was to reflect what was happening then, and to hold a mirror in order for people, like, you know, what we were talking about, to make visible these things that are invisible, in order for us to understand what is happening right now, you know, something that is a little bit more visible, a little bit more heightened or dramatic, in order for us to really kind of capture what is happening at the moment. But I was thinking about this kind of ideology of like purity or cleanliness, you know, around like these what we're talking about the pores or boundaries, and how we have the same sense of time in a way, that like the past is like there, and then the future is there, and then we're in this bubble of the present. And that, you know, there's that the same kind of ideology. It also pervades the way that we think of time. So it's really interesting for me to think about time in relationship to these other notions that we're talking about around, you know, pores and boundaries, and how we can kind of escape that in a certain way.
Right, and it is also this construct, right? I think the only thing that hasn't changed about time is the 7-day structure, but that was also arbitrary then. There's no real reason other than each day of the week was named after a different planet or god, whenever that was invented, right? So there are these structures that we still find ourselves in that we don't necessarily question. And Elizabeth, I was, you know, been continually fascinated about your project with Storefront for Art and Architecture, which is this idea of, it'll be our next question too, but living with viruses, living with microorganisms in different ways. But I think your work also has something to do with almost anticipating, if not laying the seeds down for a real change in our built environment, whether that is a common space or domestic space. I'm wondering if you can talk a little bit about that. I think you're muted.
Yeah, absolutely. Yeah, I think that the idea, you know, of living with, or like making kin, like Donna Haraway would say, is a really interesting, it's a really challenging idea when you exist at completely different scales than the thing that you're trying to get a feeling for. So as you know, as y'all were talking about before, the, you know, biological timescales are very different across, say, humans and microorganisms. Like the, you know, generation time for a microbe in optimal lab conditions is 20 minutes. So that means that in the time that we're spending here together, there will have been 3 generations of microorganisms already gone through. And sometimes that feels very fast. Like, for example, the ability of microorganisms to adapt to changes, but also sometimes it feels really slow. So, for example, you know, in the canal now, people are now the Army Corps of Engineers is coming in and just dredging all of the toxic sediment because developers want to build waterfront properties in front of it. And so I think that you know, the process of living with is also a process of finding ways to interact and finding a way to bridge those different scales at which we exist. So, you know, the scale in space is one scale, scale in time is another scale. And I think that also that comes with like a need for a certain sense of like humility and willingness to wait for things to happen. You know, in the kind of biotechnological paradigm at the moment, we're trying to treat living organisms like machines and trying to, you know, engineer them in ways that they behave just as predictably as the machines that we build. And I think that you know, the process of kind of living collaboratively or making kin with is accepting kind of the randomness and the stochasticity in these systems, and being willing to kind of put aside our like engineering approach when we're trying to work with living organisms.
Right. And, you know, with that living with idea, this is maybe a large question and we can skip it, but I am genuinely curious, like in the scientific way, other than being brought down to our knees, like what is something that we can really learn from this, from viruses or from microorganisms at all? Like how can we, you know, look at the patterns and look at the evolution and learn something from it?
Yeah, I mean, I think that that's useful for us to look at the evolutionary time scale to be able to learn these lessons. In the immediate, you know, we feel like our lives, our societies, our economies have been completely crippled by this viral agent. And it's kind of hard to see how there could be anything good that comes out of this. But I think that if we look at the evolutionary time scale, we can see that these kinds of kind of drastic mutagens are the ones that are responsible for also drastic genomic innovations. And so, like Sean, you know, when you're talking about, you know, the radiation gardens on the scale of the life of an individual species, that kind of event is likely detrimental because you'll generate mutations that are detrimental and that will kill the plant. But on a longer time scale, just by like throwing that dice enough times, then you end up getting something that's really innovative and that is adaptive. And so it's hard to think about how, you know, the coronavirus can be helping us in adaptation at the moment. But I think it's interesting to look at even the structure of the human genome itself. And so the human genome over eons has actually integrated a lot of viral structures that are then maintained as what we call transposons. So those are like at least a particular class of transposons act similarly to viruses in that they can make copies of each other and kind of bounce around. The only difference is they don't exit a cell like a virus does. And those transposons have been responsible for some of the major innovations in evolution adaptation. So, for example, the adaptive immune system. So the immune system that allows us to adapt to new threats and make antibodies to coronavirus, for example, like that particular mechanism is derived from transposons. And so maybe we can you know, gain perspective from the evolutionary time scale and see, you know, what that would mean for our species in the long run, even though on both, you know, the personal, you know, local, community and global scale, this has been really catastrophic.
But at the same time, I mean, it has, on a social scale, it has revealed some things that were really important for us to understand that have not been made visible. Social inequality, you know, how this is affecting people of color disproportionately. Yeah, and of course, like you know, kind of talking about these metaphors of kind of disease. Of course, that is revealing the constructed environment that we have and the fragility of that, right? Of these connections that we have with one another, and revealing the true labor that goes behind what is driving our society, childcare, you know, teachers, all of these kinds of things. So, you know, you could probably make some parallels to an organism as a society. I kind of hesitate to do that at times, but there are definitely some similarities of what's going on. There was, I'll leave it at that. There was another thought, but it's escaped me now.
I wanted to ask about perception, or Sean, did you have something?
Oh, I was just a small kind of thought about something that has, I guess, been gained from the coronavirus on the side of, I think there's been a real kind of breakthroughs in vaccine technology and deployment, especially with, I find that the mRNA vaccines particularly interesting, and I think there's a lot of parallels with like some of the themes that we're talking about, because the mRNA vaccine, it's basically taking some of the virus DNA and not the DNA, rather the RNA, which is what your cells read to form proteins, right? And so rather than going through the whole process of creating a kind of weakened type of virus that you're injecting, it's just the basic part. It's just the RNA, the mRNA, and then your cells take that. They start to build the proteins themselves, the spike proteins that everybody's heard about themselves, and then the immune system can recognize that. So it's kind of like a real shortcut in terms of getting the immune system to recognize a kind of, I guess, foreign entity, for lack of a better word, in the case of the coronavirus. Yeah, I think it's quite interesting, because also at that moment that the mRNA vaccine is working, it's essentially creating a hybrid cell, at least in a very small way. That cell is kind of wearing some of the virus's outer shell. And so, this kind of, it goes back to a little bit this porosity between humans and viruses and all kinds of organisms and other organisms, in a sense. Anyway.
No, that's great. And I was also thinking about the spike in the molecule too, and the receptor binding variations, which is such a hard project to communicate to someone over a screen, since most of the work is scent based. But to give maybe like a small anecdote for those who haven't been able to be in a room with those works, there are these scent diffusers mounted on the wall, but the molecules that you've created are scents that never existed before, like you said, new colors, like seeing a new color, but you can't see a new color because we only have so many cones in our eyes. Right? So what the experience of that scent does is it goes to the close, and you can correct me if I'm wrong here, it goes to the closest familiar scent that someone might know. So I remember there being like the same shared balance between like a lime and a cherry, but your scent is like somewhere in between those two things, maybe using the wrong fruits or citrus. But if you smell it for one person, it'll smell like cherry. For the next person, it'll smell like lime. There's all of these, the misfires, which is, I think, to Elizabeth's point, we've treated our bodies like machines in this new era and almost left no part for the very real margin of error that is the human experience. Things get categorized in ways that are familiar. Okay, well, we have a lot of questions from the audience. I'm going to ask one before I ask a few more of mine, and then we'll do the rest at the end. So viruses and microorganisms are so small, yet their implications are huge. How do you balance this enormous scale shift scientifically, socially, etc., in your practices? I guess it's another version of time, but for size. Who wants to take?
In the way that I'll take that first. I mean, the way that we react to it is very visible and very human scale. And there's an aesthetics to the way that we have reacted. I mean, everybody is so familiar now with PPE, right? Like the bunny suits and the shields and everything. I mean, the history of viruses and microorganisms is so interesting because it's so invisible. So we've had to deal with this invisible thing, and the stories that we've told about those things have like changed throughout time. I just was thinking about a story about an ant who has been kind of like, unfortunately, like politically not, on my side, I feel like, but has been traveling a ton, and she was like, oh yeah, I just go, I felt totally comfortable going to this the restrooms at this the Cracker Barrels when I was traveling. They look totally clean, you know, and it's kind of perception that we have of cleanliness and how that has jumped to a social scale. Perception that people have of their own cleanliness or of a group of people's cleanliness. I find that to be very fascinating. And those are the things that I, as an artist, I observe, and they go into the work in some way. Sometimes they end up being something that it becomes part of the work. Sometimes more about like a kind of general feel for what I'm observing about the world at that time. Thanks for the question.
Well, and you touch upon something which is this perception and the relation to the instrument, which Elizabeth, I think you first brought up, which was, you know, how do you see these things? If you can't see them, if they're too small for us to see, how do you see? And I'm wondering how your relationship to the instrument, which I guess in your case is a microscope or various, you know, things used in a lab in order to see the unseen. How has your relationship with it changed over the years? You know, with new technology coming in, have you seen a shift?
Yeah, I think it's interesting to acknowledge that there's maybe two categories of instruments that give us those interfaces. One of them is the ones that were explicitly designed as such, so scientific instrumentation, for example. And then other ones which are maybe more vernacular objects or practices that do mediate an interaction with the microbial world, but were not necessarily designed as such. And so for the first category, I think like when I started out, you know, in my career as a scientist, and I was like, okay, cool. You know, these instruments are like extensions of our senses, and they give us kind of ground truth information, like unbiased data. And as I've, I guess, matured as a scientist, I realized that all of those instruments are designed under a particular paradigm, and therefore carry the particular bias of that paradigm. So you design an instrument to perceive a particular like range of frequencies because you think that that's the important range of frequencies, but by definition, you're omitting what is not within that range. You're not able to see what's in that range. And so I feel like I've gone through a few, there's like fads in biological research where there's like this like new tool that comes out, and it's like, yeah, once we know this, we'll know everything. And so, maybe 15 years ago, it was microarrays. So being able to measure the expression of genes. And we were like, sweet, like once we know how to measure the expression of genes, we'll know everything about everything. And there'll be like all these things that we don't know will finally be elucidated. And then like 5 years after that, it was like epigenetics. And we were like, oh, wow, we can measure like epigenetic modifications in molecules. Like that's going to explain everything that we don't know. And now, in the last maybe decade, it's been DNA sequencing. And we're like, sweet, once we can sequence all the DNA, which is, the like intrinsic information storage molecule, we'll know everything about these organisms. And that's still not the case. There's still so many mysteries that remain. And so I think my relationship to instruments has been one of like increasing skepticism, maybe, or not exactly skepticism, but increasing acknowledgement of that they're designed with a particular thing in mind, and that comes with a set of biases.
Or is there comfort? Is there comfort in the unknown? Like at least knowing how much you don't know?
Yeah, that's what keeps it interesting for sure.
Yeah.
I was wondering if I could ask Sean.
Yeah, I was going to bring it to Sean, but you can go.
Yeah, I was wondering if I could ask Sean a question that maybe like extends. I don't know, maybe it's a little bit off. But you were talking about like kind of biodiversity. I'm wondering if in that conversation you are factoring in indigenous knowledge about biodiversity and whether like the time scale of that, whether that factors into, whether the practices factor in, whether they're concepts of time or connection with those plants factor into your thinking.
Yeah, well, I think that definitely like there's I think a lot written about how indigenous cultures have been much better stewards of ecosystems, throughout the world. And in a more specific to my own project, I would say to some extent I've looked into that, but I don't think that there's. I mean, I wish that that kind of knowledge, was more maybe represented, in the field. But yeah, it's something that I'll probably look into further. There is one thing that I think is relevant to that, though, is this. There's just this one particular species that that has been in the. So I live in Michigan, in Detroit, and so in the region there's a native species called the pawpaw, and it's kind of similar to like something between a mango and a banana, and people don't eat it. It's like basically since white settlers came through the region, no one's really eaten them, but they grow like throughout local forests, and that's actually one of the focuses of this particular, well, the first kind of test plot that I'm working with is to include those, because it also provides food, and so yeah, anyway, there's kind of a long story, but the local area around there can go into the forest and take any food that is produced by the trees and things like that. So yeah. Mm-hmm. But one thing back to the to this question about kind of smallness, or how to see these kind of small things, or how to deal with not being able to see them. In the case of, that's part of what led me to being very interested in like molecules and the kind of shapes of molecules, is that without any instrumentation, you already have like a direct inbuilt way to experience them, which is the sense of smell and taste in some cases. And so, yeah, I just think it's kind of an as instruments of the body go, it's a kind of underutilized instrument. Obviously, much of what scientific discourse studies is something that can be visualized in in some form. But yeah, I think there's a lot that's kind of there in the kind of sense of smell, and what can be experienced and learned that way. That's not that doesn't make it into discourse, I guess.
And Hung, I just wanted to pick up on, sort of the importance of thinking about Indigenous knowledge when talking about each of these projects too, right? Like the 4 tenets of adaptability, cumulative knowledge, dynamism, but also like a holistic nature. I think that's what we're really getting to in all of your works, is that, I guess it's close also to Elizabeth's description of an instrument. You can't just focus on this one part here. It doesn't tell you the information until you open up the spectrum and sort of consider all of the possible factors that go into why something is the way it is, which is certainly how, Escape was able to be made 4 years before a global pandemic hit, right? Because you're
a long time ago. Yeah.
Was it that long ago?
2004.
Okay, yeah. But really opening up our eyes to see that everything that's happening now is part of a larger system, which doesn't make it all the more palatable or easier to digest, but there is some comfort in history, unfortunately, repeating itself over and over again. So we have 2 more questions from the audience, and is it okay if I go over a minute, 2 minutes? I'm gonna go over a few minutes. Question 1. Exchange appears to be a theme in all of the artist's work. Exchange between the human body and the environment, exchange between humans of different cultures, exchange between humans and microorganisms. What exchange do each of you hope to have in a post-COVID world?
I'm just excited about like going to a restaurant and eating with friends, exchange with people. I mean, we're all collaborative artists and we are not able to be with people.
I think what I'm looking forward to is unexpected interactions. I feel like now all of my interactions need to be planned. You know, like I plan to speak with a friend or colleagues or whatever, but I realize what I really miss are the unexpected interactions of just like being out and about in the city and having random conversations with people.
I don't have anything to add. I think that covers it. Both of those answers cover it very well.
At least the
If
nice
we have just one more.
I was just going to say the nice thing for me has been spending time with your work where, you know, finally some new information has entered into my sphere, right? There's the calculated randomness of some of it has been helpful and healing in this time of not being able to see all that much that's new, but to be able to think about a lot that you haven't had the time to do before. So I wanted to thank all of you. I mean, any closing remarks? We need to wrap up now.
I think maybe just following on that thread of Indigenous knowledge in the context of our practices. And I think, you know, we think of biotechnology as being a really like recent discovery of the Western world. But actually, you know, we've been interacting with microorganisms in really deep and meaningful ways for eons. And I think that, you know, reserving the word technology for recent developments in that relationship is erasing the fact that there are indigenous technologies, very explicit and intentional technologies that have been designed and used to interact with microorganisms and the invisible biological component of our world for a very long time. You know, the kind of most relatable example being the practice of fermentation, for example. So, you know, creating the vessels with the shape and the materials necessary to promote those kinds of interactions. Or also practices of agriculture, like, you know, planting the three sisters together, which actually is an amendment to the soil microbiome. And so I think, yes, I think we need to like, you know, relinquish that hold on the term technology as being a recent Western thing, and acknowledging that those technologies have been developed by Indigenous peoples for a very long time.
Thanks, Elizabeth.
Mm-hmm.
Yeah, and I think each of your works also does, you know, really open all of our eyes to things that need to be talked about. And I just wanted to thank you for all of the work that you've done over the past years, and having the chance to talk about it with all of you and share with our audience here has been really amazing. So again, the live transcript will be published on Ocula, albeit with a few edits. But thank you so much to everybody who joined, and see you at the next program for Alternate Assembly. Thank you all.
Thank you so much.
Bye.
Bye.